Welcome everyone. Thank you so much for having this fire chat about a very seminal paper that was published recently in the Journal of Chemical Science about protein fold and AlphaFold use in application of AI also in the drug discovery using Chemistry 42 and other platforms of Insilico Medicine, which is very a seminal paper, I believe, for the entire line from the biologists and chemists to AI computational scientists and clinicians like myself. And here are some of the co-authors. So I'm extremely excited to talk about all those aspects with you and discuss the impact of the paper and the importance of the paper for the entire field and of course for the real world, for the patients. And I would like to start with a question to the co-author, Professor Michael Levitt, exactly opening with the significance and the importance of the paper, perhaps maybe really with the focus on what's so special and unique about applying those two AI platforms, Chemistry 42 and all the pipeline of Insilico and AlphaFold, to create a new target, identify it?
Well, that's a really good question. Basically, the field of protein structure underwent this revolution about 2 years ago when AlphaFold was able to solve, or at least provide models for essentially all the sequences that existed. Some of these models are better than others. But having a model, it depends what you use the model for. And quite frankly, no one believed that the models would be good enough for drug design. In fact, there was a paper in Cell where they actually looked at 20 AlphaFold models, tried all the regular programs for drug design, and showed that none of them actually really work. So the key thing about the Insilico Medicine paper is that it worked. And I think part of this is because one of the key things about Insilico is that their pipeline is optimized from the beginning to the end. So most people can say it doesn't work because it doesn't fit. But Insilico can say it works because we found a protein, we found experimentally that this particular drug works. And there's nothing better test than that. That's something, though, that hardly anyone can do. So essentially, for people who were looking at this large mass of structures, knowing that it can work, I think is really, really important. And I think Insilico, it's not clear that others can get it to work yet. We still haven't seen that. But we have seen in these examples that this vast wealth of structure can lead to novel drugs, and that's a really, really important step forward.
Thank you so much. So it's really a revolutionary step, opening up a door to yet another level of AI drug discovery, if I may ask, right? Dr. Alex Zhavoronkov, the senior author of the paper. Could you expand exactly on that? What is the impact of the AI drug discovery? How those two platforms worked together? How was the symbiosis created here? And what are the future developments, especially in that sense?
So sure. Well, before I go into that, I just want to compliment DeepMind on their achievement in terms of creating AlphaFold. That particular piece of work got a lot of public attention and is probably the most publicized advance in artificial intelligence for drug discovery. So everybody is talking about AlphaFold. And again, deep bow to Demis Hassabis and his team for making not only very significant advances in deep reinforcement learning from the very beginning, but also steering it towards drug discovery and popularizing it. So we actually try to learn from DeepMind in many ways, also in terms of how they publicize things. Of course, we would not be able to achieve this kind of PR capabilities that they have, but we also want to ensure that we inspire a lot of scientists, a lot of young scientists, people in the industry, people with a lot of experience, to try deep generative reinforcement learning and trust it. So currently, this technology lacks trust. So what we wanted to do is we wanted to take the AlphaFold protein from the repository, public repository that AlphaFold constructed, plug it into our pipeline, and go from the beginning to the end, as Dr. Levitt said. And at that time, this paper is now 1 year old, actually more than 1 year old, because we put it on a preprint server in January, February 2022. So it took us almost a year to publish. So back then, we decided to do a race and go from beginning to the end using our PandaOmics target discovery platform to identify the target that lacks a crystal structure, is not exactly difficult to drug, and at the same time can play and be very highly ranked in hepatocellular carcinoma, in HCC cancers. And then we used Chemistry 42 to identify the pockets on this AlphaFold structure. We had to modify it a little bit, so very few structures are usable right off the bat. So we had to modify a little bit, throw it at Chemistry 42. Chemistry 42 consists of 42 generative engines. Some are GANs, some are genetic algorithms, some are transformer neural networks, and they start generating many, many molecules in response to the crystal structure they have. And we have the reinforcement learning platform consisting of a reward function with 500 different pre-trained models that evaluate the output of each generative model and see if the molecules are good or bad, and very rapidly either reward or punish each generative model and the ensemble as a whole. This process takes 72 hours, so it's not as snappy as ChatGPT, but there we don't need snappiness, we need accuracy. And we got very quickly to just a few molecules to synthesize, tested, got micromolar hits, respectable hits, optimized again using now ligand-based drug design, and got to respectable nanomolar hits. So it's still not a drug, but we demonstrated that it works, and it works all the way into human cells. And we tested it, and we did it very quickly. So the entire process is like 50 days, which is pretty cool. It's basically under 2 months. And from the beginning, it's 30 days to a very low level hit, and then to a strong hit another 20 days. So that process shows you it's not something that we are taking into the clinic right now, right? So not the drug, but as a demo, it's a very good demo showing that AlphaFold has value, but also that it's a piece of a bigger drug discovery puzzle that you need to construct. If you don't have every piece of the puzzle, you won't be able to move so quickly. And also you need to have experienced drug hunters and preferably super experienced medicinal chemists like Dr. Ren and Ding Shao, who have many years of experience and who can very quickly process the output of all of those AIs and turn it into actionable projects. As a matter of fact, just recently, and very serendipitously, one professor approached us from the China University of Hong Kong, Albert Cheng, and he came to us. He's also in Hong Kong Science Park, so we never realized that we have somebody of that caliber working in the Hong Kong Science Park, and also he is a vice dean of the China University in Hong Kong. And he told us, look, actually I've been working on CDK20 in HCC for a very long time. And we looked up, and he is like the top key opinion leader in that field. So, who knows, maybe we will actually progress that project into the actual clinical program. So we could collaborate, but that requires, I guess, a little bit more polishing right now.
Thank you so much for expanding on those processes, and thank you also for bringing us to my next question, which is medicinal chemistry and the impact of the experts in the team. So in many of your interviews, you are mentioning how the multidisciplinarity of the team is so crucial for the success. So it's not only the AI and the entire pipeline from A to Z, like Professor Levitt mentioned, but also the team, right? AI computational scientists and medicinal chemists, biologists, and I know that in the huge network of collaborations, you have plenty of clinicians, which from my perspective is one of the main things also to keep everything running. But coming to you, Dr. Xiaoding, thank you so much. And congratulations also on being co-author on the paper. I would be extremely interested to learn what is the perspective, what is the role of a medicinal chemist, right? A lot of people don't even know what exactly is the role here, especially in such a progressive team. And perhaps you can focus a little bit on the validation, right? On the target that was identified.
Okay, thank you for the question. So I think in general, from the role of the medicinal chemistry in the AI generated model, I would like to talk about the Chemistry 40 role in our daily life. I think for me, my own experience is that Chemistry 42 is kind of like my talented colleagues surround me. We discussed, I think we communicate every day and we learn from each other. So using a CDK20 as an example, I think we find the right target, right, for the HCC, and we know that from a dark target without a small molecule inhibitor and without protein structure, and we use the AI tools using the AlphaFold predict structure and to generate a pocket. And then the Chemistry 42 generate thousands of molecules to us. Then we need to pick the best molecule from the thousands of molecules, and we need to have the sense which molecule will be, have a high possibility to produce, and also if there's a potential, some alerts and some, high potential. So we need to select that. And I think the very interesting thing that we select the 7 molecule, and we synthesize the molecule, but that doesn't mean that all the molecule will have the potency. And we very fortunately, I think, one of the 7 molecule demonstrated very good activity, and then we give feedback to the Chemistry 42, and we, like what I just said, we communicate, and then it gives us more potent molecules. So I think there's not only for CDK20, we have lots of in-house programs that we, kind of back and forth lots of times, and it will give us lots of good molecules to proceed. So, from this actually very successful example of CDK20, we later will have more publications on the collaboration. I would like to say collaboration from medicinal chemist and Chemistry 42. Yeah.
That's extremely exciting.
Yeah.
Thank you so much. And I believe it's great to learn how not only the communication between the team members is evolving and bringing something so extremely powerful, and we all know how difficult it is to understand the language, right, between specialists in computational sciences and biology and so on, life sciences, but also the communication between AI and the human, right? So, so that's extremely fascinating. And as you just mentioned, over 900, or almost 900 potential targets identified, and then selecting them all down, right? Really giving the hunt and finding the right way on identifying further the toxicity and druggability. I think that's a fascinating process, and I know that Dr. Ren Fang is extremely high-level expert in that field. So I'm extremely curious to learn from you specifically for this work that was done, and perhaps more of an overall perspective. What does it take to take it from the hunt of the target to further evaluate all the other aspects which are important in order to finally say, okay, this is a potential target of druggability, and to then take it further?
Yeah, I think for this case, we might not be able to take this target, CDK20, all the way to the drug, because there's a lot of unknowns in biology, because this is a very novel target. We need to figure out the biology MOA first. Maybe we can collaborate with the professors in a Chinese Hong Kong University, right? So that's one possibility, but at the current stage, we might not be able to take this particular target all the way to the drug. But for a lot of other targets, which are also novel, for example, our program 1, which is a very novel target, and we have successfully progress the target and the molecule all the way from target identification to the clinical trial, phase 2 now. It took us a lot of effort of doing this, right? So we spent a lot of time. We spent 18 months to identify the targets, validate the target using like CRISPR and other technologies. We discovered the small molecules with potency. That's the first step. Then developability, then efficacy in animal models. Then we need to further validate the efficacy in the human cells, human IPF cells, and then DMPK, and then safety. So there's a lot of several hundreds of experiments we need to do before we can nominate a PCC, a single PCC. That's a tremendous work, and that also demonstrates Insilico's spirit of doing additional works to make sure our drug is safe and efficacious. So I think from our point of view, to take one target from the very beginning all the way to the clinical trials, we have keep very high standards of our criteria of nominating the PCC, and also we keep very high standard of the criteria to progress from PCC all the way to the IND. And at the same time, we keep very high standard of criteria in the clinical trial development, especially in patients. So for AI drug discovery, we have two different philosophies. The AI, we need to be fast. We want a very fast speed, right? Mm-hmm. For drug discovery, we need the compound to be safe, and it need to be compliance with all the regulations, right? So now we want to merge these two different, spirits into one company, and that's Insilico Medicine. Mm-hmm.
That's really fascinating, and I really appreciate especially the thing you mentioned. Safety starts at the PCC level. Right? Not only as for us, safety first for the patients. Mm-hmm. And thank you also for pointing out something that brings me to my last question to Professor Levitt, if I might, about the scalability and the future of this type of AI drug discovery and an augmented generative AI implementation in that sense as well. What do you think are the opportunities, perhaps challenges? Where do we stand?
I think in terms of scalability, which is basically be able to handle multiple projects at the same time, I'm actually really impressed by Insilico Medicine because, they are still a relatively small company compared to Big Pharma. And Big Pharma often struggles to have more than one target ongoing. Difficulty with designing drugs is it's a numbers game. You cannot know at the beginning exactly what the success is going to be. So the whole use of AI to basically make the whole process smooth and try to evaluate as much as possible what the success is going to be, I think is remarkable. I always have a feeling that in the company there's real synergy between the different projects, the different targets. So I am extremely impressed about this. I think the experience with the AlphaFold model is doing something that's actually very, very difficult, not just because it's a protein structure that's a model, but if you've solved the structure by crystallography, you almost always will have the structure of some inhibitor bound. Mm-hmm. So now you're starting out with a model, but no idea what binds to it. And this is what was done here. So I think that this really is a demonstration of much greater capability. It's also sort of interesting that it was almost done as an exercise. So in order to have the internal energy to do that is really impressive. So I am actually, again, each time I discuss with Insilico, I'm actually more and more impressed. I think we have real potential for scalability. But obviously, I doubt if they're suddenly going to jump to 1,000 targets. There's logic here. But I think if sticking to the idea of rapid AI and rigor when it comes down to all the other steps that involve human beings is incredibly important. I do believe that it's soon it will be a AI model of a human that's part of Insilico, so that the human aspect will be less and less surprising. I mean, it shouldn't be, oops, I didn't know that would be. There must be a way of, in some ways, putting all that together, either using general large language models or generative AI, and I'm sure they're really thinking of it. We haven't had a chance to discuss this, so I'm extremely excited.
Wonderful. So am I, and thank you for bringing out this perspective to us. Thank you so much, everyone. I just, if I may, would like to add from the clinical perspective, from the very end of the chain line, that it's a dream come true for any clinician and also patients. Many patients turn to us and they actually ask, they follow what Insilico is doing, or AI-based drug discovery, and they are asking, is there already a way for us to use a drug or to help to create a drug for something that is as difficult as HCC, right? The 5 years prognosis is extremely poor, and we have barely any drug that is working, and many other diseases. So I believe this type of a pipeline, AI from A to Z, and really the implementation of the newest level of AI, but always putting the rigor of safety and efficacy, is something extremely promising. So we're very much looking forward to implementing not only the targets, but also the pipelines in the clinic in the near future.
Thank you very much, and let's achieve more together. Thank you.
Thank you.
Thank you.